The materials needed for this technique are a pipette aid, chem wipes, DMEM, media, transformed mouse fibroblast cells, and an adherent flask, additional tissue culture flask, waste beaker, trypsin, sterile pipettes, and 7% ethanol, bottle, or spray. To maintain sterile technique, wear gloves and disinfect them with 70% ethanol. Disinfect the laminar flow hood as well and wipe the surface with chem wipes. Obtain a flask of fibroblast cells and assess the confluency of the cells in the microscope. After disinfecting the materials and hands before lab work, use a sterile pipette with the pipette aid to remove the old media from the flask. Then remove the pipette into the waste beaker. Next, use a sterile pipette to gather 2 milliliters of trypsin from the conical tube. Then transfer the trypsin into the flask. Trypsin helps detach the adherent cells from the flask. Remove 1 milliliter of trypsin after 10 seconds. Gently tap the flask to ensure all the cells have been detached. Then place the flask on the microscope and check to see if the cells rise. Pipette 2.5 milliliters of media and dispense it into the flask. The media deactivates trypsin and is used to feed the cells. This brings the total volume of the flask to 3.5 milliliters of liquid sample. Next, grab a microcentrifuge tube. Pipette 0.25 milliliters of liquid sample of cells. and dispense them into a microcentrifuge tube. Next, determine the cell count. Use a pipette man and set it to 10 microliters. Take a 10 microliter liquid sample of cells and dispense them into a new microcentrifuge tube. Grab TC10 trypan blue dye solution and take 10 microliters of solution. And gently pipette it into the 10 microliter liquid sample of cells. Mix sample by doing resuspension a couple times. Grab a counting slide. Dispense the blue dye sample mixture to one side of the chamber of the counting slide. Place the chamber slide on the side with the sample into the electronic cytometer device. This detects the amount of live cells from dead cells in the sample. We have enough cells to passage into 2 flasks. After doing the cell count, we can add the appropriate amount of media to the flask. In this case, 3.5 milliliters of media is added to the existing flask. This brings the volume of the flask to 7 milliliters. Next, we take the 2 flasks and prepare them for passaging. Pipette 7 milliliters of liquid sample of cells. Resuspend the liquid sample of cells between 5 to 10 times. Add 3.5 milliliters of liquid sample of cells to the flask. Then add the other 3.5 milliliters of liquid sample of cells to the other flask. Be careful to not dispense any bubbles left over from the pipette to the flask. Obtain the new passage tissue culture flask out of the fume hood. Place the tissue culture vessels on face down. Move them side by side to make the liquid sample spread out in the vessel. Then label the tissue culture vessels with the passage number, name of the sample, and date. Attach the two flasks on top of each other and place them into the incubator at 37 degrees Celsius. After doing lab work, turn off the pipette A, put away materials, and clean the hood.